Hello, science enthusiasts. Welcome to the Fantastic Datasets channel. Today, we'll explore how researchers at Google DeepMind solved a 50-year-old challenge in biology, the protein folding problem. Before we get into details, let me show you the DeepMind blog on AlphaFold. This webpage is packed with fascinating insights into this revolutionary innovation. Why is this important? What a protein does depends on its unique 3D structure, and understanding these structures is key to solving biological puzzles, like drug discovery or understanding diseases. The challenge of figuring out protein shapes is known as the protein folding problem. So, how do we predict the shapes of these tiny machines in every living cell? That's where AlphaFold, Google DeepMind's groundbreaking AI, comes in. It has solved a problem once thought unsolvable. In today's exploration, I'll explain why AlphaFold is so important and walk you through this incredible achievement. But first, what are proteins and why does their shape matter? Let's find out with this short video from the AlphaFold website, which perfectly explained this groundbreaking science. Let's watch it together.
Inside every cell of your body, billions of tiny machines are hard at work. They carry oxygen in your blood, let your eyes detect light, and even make your muscles move. These machines are proteins, and they underpin every biological process in every living thing. Each protein has an intricate 3D shape that defines what it does and how it works. We know of over 200 million proteins and counting, but we only know the exact 3D shapes of a fraction of these. Unravel a protein, and you'll see it's like a string of beads made up of 20 different types of amino acids. Interactions between these amino acids make the protein fold, as it finds its shape out of almost limitless possibilities. For decades, a community of scientists have been working on a way to figure out a protein's shape, just from its string of amino acids. This is a major challenge in our understanding of biology. So we created an AI system to solve this problem called AlphaFold. It's trained on the sequences and structures of about 100,000 proteins, painstakingly mapped out by scientists around the world. Today, it can accurately predict a protein shape just from its sequence of amino acids. AlphaFold's predictions could enable progress in all sorts of areas. Imagine a future where we can understand diseases more quickly and develop drugs to fight them, or one where we could use enzymes to break down plastic waste, or even to capture carbon from the atmosphere, all with the help of proteins. There's a lot more work to be done, but unlocking the shapes of these building blocks could help scientists better understand the natural world, and perhaps expand our knowledge of life itself.
Before starting to explore how AlphaFold solved the problem, it might be interesting to know that researchers have successfully determined protein structure experimentally, using methods such as X-ray crystallography, nuclear magnetic resonance spectroscopy, and cryo-electronic microscopy, but these methods are time-consuming and expensive, and that's why the structure of only a tiny portion of known proteins was determined. Now let's see how AlphaFold algorithm works. Here is a simplified diagram of AlphaFold 2's architecture. We'll break it down into 3 sections and start here when an input sequence of residues, or amino acids, is entered. AlphaFold compares it to several databases of protein sequences to extract similar sequences from various organisms and tissues to generate a multiple sequence alignment, or MSA, and an initial MSA representation. It also pairs the input sequence and searches databases for templates of similar sequence proteins with experimentally determined structures. This is then used to create an initial pair representation of the input sequence representing the relationship between every pair of residues within the target protein. From here we can move on to section 2, the evoformer. The evoformer is a neural network unique to AlphaFold. It consists of 2 towers that can communicate information to each other. For the MSA representation, the neural network prioritizes looking for row-wise relationships between residue pairs and the input sequence before considering column-wise information that evaluates the importance of each residue in context of the other sequences. The pair representation tower evaluates the relationships between every 2 residues, which can be thought of as nodes, to refine the proximities or edges between the two. It achieves this by triangulating the relationship of each node in a pair relative to a third node. The goal of this process is to help the network satisfy the triangle inequality theorem, where the sum of two edges on the triangle must be equal to or greater than the third. So how and where do the two towers communicate before the pair representation calculates the triangulations on each edge? It considers the updated MSA residue relationships and updates each edge accordingly. The results of the pair representation data is then used in the MSA's row-wise weighting of pair relationships in the input sequence prior to its next round of row and column-wise evaluations. These individual and cross-communicated calculations happen 48 times in the EVO former before creating the refined models of the initial MSA and pair representations. The final section involves another neural network called the structure module. It takes the refined models and performs rotations and translations on each amino acid, revealing an initial guess of the 3D protein structure. It also applies physical and chemical constraints dictated by atomic bonds angles and torsional angles. The refined models, as well as the output of the structure module, is iterated back through the EVO former and structure module process 3 more times for a total of 4 cycles, before it arrives at the final result. Predicted 3D atomic coordinates for the protein's 3D structure. AlphaFold's remarkable accuracy is one of its most fascinating aspects. Let's dive into the results of CASP14, the gold standard for protein structure prediction models. In CASP14, AlphaFold achieved an unprecedented median global distance test (GDT) score of 92.4 across all targets. GDT measures how close a predicted structure is to an experimentally determined one, with 100 indicating a perfect match. A GDT score above 90 is typically comparable to experimental methods like X-ray crystallography. In fact, AlphaFold's predictions for many targets were so accurate they achieved atomic-level precision, once thought unattainable by computational models. Why does this matter? This accuracy unlocks the potential to solve critical biological challenges. Having this background about AlphaFold, now it's the time to explore the AlphaFold Protein Structure Database website, a powerful resource for studying protein structures. Developed by DeepMind and EMBL-EBI, it offers open access to over 200 million protein structure predictions, covering almost all cataloged proteins. Whether you're a researcher, a student, or just curious, this tool makes cutting-edge protein research accessible. Its powerful search engine allows you to find specific proteins by name or UniProt ID. For example, searching hemoglobin subunit alpha using its UniProt ID, P69905, brings up an interactive 3D model. You can rotate and zoom to explore its intricate details. If you want to download a protein structure, simply click the Download button to get the structure file for further analysis in relevant softwares. For large-scale studies, the bulk download option allows access to the entire dataset. What makes AlphaFold especially noteworthy is its reusability. As all source codes and data are openly accessible, anyone can use them for diverse applications. This aligns perfectly with the FAIR principles, ensuring the data is findable, accessible, interoperable, and reusable. This openness accelerates global collaboration and breakthroughs in science. It's also important to understand the licensing conditions. The contents of this database are released under the Creative Commons Attribution 4.0 International CC BY 4.0 license. This means you are free to use, share, and adapt the data as long as you give proper attribution to the creators. Be sure to review the full licensing terms here to ensure compliance with your intended use. In summary, the AlphaFold Protein Structure Database is a groundbreaking tool for exploring, downloading, and applying protein structure information in your work. There is a point that you have to consider. While AlphaFold is revolutionary, it has limitations. It struggles with low confidence regions. areas of proteins that are naturally disordered or lack a stable structure, despite their critical biological roles. AlphaFold also faces challenges with protein-protein interactions, large complexes, and ligand or membrane interactions. Additionally, it predicts only static structures, while proteins are dynamic, constantly changing shape. Another challenge is that its accuracy declines for novel or highly divergent proteins with limited similar data and doesn't account for real-world factors like pH or temperature, etc., which influence protein structure. Despite these challenges, AlphaFold's impact on science is remarkable. It has transformed how we study proteins, making it worthy to win a Nobel Prize, as its achievements redefine possibilities in biology. With that, let's wrap up our journey of exploring AlphaFold here. Thank you for joining me as we uncovered the science behind this groundbreaking tool. AlphaFold has not only solved a challenge that puzzled researchers for decades, but also opened up new possibilities in biology, medicine, and beyond. If you enjoyed this video, don't forget to like, share, and subscribe to the Fantastic Datasets channel. There's so much more to explore in the world of science and data. Until next time, stay curious and keep exploring.